Pat Callie's talk, as soon as I find my paper here, is on, essentially he's going to talk to us about what we can learn from the carnivorous New World pincher plant. So, Pat? It's going to take me just a second.
My group is interested in plant evolutionary biology, and one of our platforms is the New World carnivorous plants of the family of Sarraceniaceae. And I'll give you guys a quick background about these plants, why they're important, our investigations into these plants, and then some directions we want to go into, which will involve a new technology, an emergent technology of next-generation sequencing. So the family is called the Sarraceniaceae. It has 3 genera: Sarracenia, Darlingtonia, and Helianthora. And the distribution is quite unique. The 3 genera are separated from each other by rather wide geographic distances. Darlingtonia in panel A is found only in the Pacific Northwest, the border of Oregon and California. Helianthora in panel C is found in South America on these plateau mountains called tepuis. There was an article in the New York Times about 2 weeks ago about some frogs on tepuis, some neat discoveries about endemism on those particular habitats. And the most widespread pitcher plants in the New World are in panel B, the genus Sarracenia. These plants are important for several reasons. First, biologically, they represent a remarkable manifestation of the evolutionary process in terms of their actual leaves. What you're seeing are leaves modified into so-called pitchers that trap rainwater. In this rainwater inside those pitchers, the plants secrete digestive enzymes. They also secrete attractive compounds. They lure insects to those pitchers. Insects will fall into pitchers, drown, be decayed by digestive enzymes, and a whole community of bacteria and other invertebrates that exist in those particular pitchers. These plants all exist in nutrient-poor habitats, and so they rely upon these captured prey to supplement their nutritional needs. Second reason for their importance is they're all really threatened as far as their existence goes. Their habitats are all quite delicate. The tepuis in northern Venezuela are UNESCO World Heritage Sites. The Sarracenia occurs mostly on coastal plain areas which are now being subject to rather rapid development. So pretty much all of them are rather threatened as far as their particular existence goes, and high priority in terms of conservation biology. An example of the more extreme habitat, the tepuis, again, these are mountaintop plateaus accessible just by helicopter. Biologically, they're often quite distinct from each other as far as their species composition. Many species of animals and plants are endemic or unique to a particular tepui apart from the others. So we chose to examine the evolutionary history of these plants. Where do they first arise in the New World? What are their relationships? And can we get some sense of the timing of their actual appearance? We utilized DNA sequences as our data set and computer models to build essentially a family tree, which you can see here, of the actual plants. Our efforts resolve relationships of species and genera, and also gave us a sense of how these plants first arose and evolved. And basically, the group arose based upon our model first in South America some 47 million years ago. It then spread throughout the continental U.S. Then these plants were subject to the effects of global climate change during the Pleistocene era. Our model suggests that Darlingtonia, the Pacific Northwest taxon, diverged first from the other species and genera about 35 million years ago. Then Helianthora, the South American genus, began to diverge from Sarracenia about 7 million years ago, with Sarracenia being the most recently diverged group. Essentially, these plants were formerly widespread across North America, but due to Pleistocene glaciation, various climatic changes, populations in the interior U.S. went extinct, leaving us our current remnant populations on the West Coast, East Coast, and also South America. We're also very intrigued by these plants for the following reason. In their pitchers exist a whole unique community of individuals. For example, there's a mosquito, Wyomia, which is found only to exist inside these pitchers. There's also a particular mite of the genus Sarraceniopus, which likewise is found only in the fluids inside these various pitchers. More remarkably, most Sarraceniopus species are unique to a particular species of pitcher plant. It's like there's been a tracking of evolution between the pitcher plant and the actual Sarracenia species. So we have these amazing ecosystems inside these particular plants. This diversity goes to within the leaves themselves. Those prior organisms were existing inside the pitchers. They recently found, two investigators, endophytic fungi living within the actual cells of the pitcher plants. Great diversity of endophytes in this particular case. And so we are now going to next generation sequencing in an effort to characterize these particular communities. UK is hosting a workshop this summer on NextGen sequencing, and the two reasons for doing this particular effort: number one, the revolution we're now seeing in biomedical science as far as NextGen sequencing will be paralleled with the revolution we'll be seeing in ecology and evolutionary biology. And in terms of developing a new workforce in life sciences and computer science, NextGen sequencing will be a major driver of that particular development. Thank you.